Thank you very much. Good morning, everybody. Thanks to the conference organizers for letting us present and all of you who are attending. By way of quick introduction, I'm Rick Modi. I'm the founding CEO for Affinia Therapeutics. Prior to this, I was the Chief Business Officer at Avexis, where we brought Zolgensma to patients with SMA. Before that, I was at Intermune as Senior Vice President. We brought Esbriet to patients with IPF, and I started out my career at Centocor, where we brought Remicade to many different patient communities. At Affinia, our vision is very simple. We want to replicate the success that Zolgensma had in spinal muscular atrophy, but to do it in prevalent diseases. The way we're doing it is through our ART platform. ART is Affinia Rationally Designed Therapies, and we believe we're addressing one of the biggest limitations in gene therapy, one of targeted delivery. With ART, we're actually applying a structure-based design approach to AAV capsids, leveraging both regressive analytics for structural activity relationships as well as generative machine learning. The platform has been applied to two tissues where we believe delivery is the biggest limiting factor: in CNS, crossing the blood-brain barrier, and in muscle. And in both these two cases, we use an IV delivery. Whereas our initial work is focused on AAV capsids, this platform may have applicability also beyond AAV capsids into other modalities, which are non-viral as well. Since we formed the company, the new capsids that we now have in hand in NHPs have shown striking improvement over AAV9. When I say striking improvement, these are 2 or 3 orders of improvement in expression in NHPs when given IV, in CNS, in heart, and in skeletal muscle, as well as at the same time detargeting 2 tissues that are significant liability, unfortunately, for AAV. This is the liver, and this is the dorsal root ganglion. The company is led by a set of leaders who are industry veterans who have brought INDs into the clinic, who have gotten gene therapies approved by the FDA, and have had significant M&A and exits for shareholders as well. Our platform is credentialed with a deal that we have with Vertex, which is now in its 3rd year, and both parties are very pleased with it. And we're in the midst of preclinical studies for our own internal pipeline with programs. We believe what we're doing with ART is actually changing the paradigm of AAV capsid discovery to something that is akin to classical drug discovery. What we start is by determining the attributes of the capsid that we need, the molecule that we need, and then we go about designing it in a very methodical manner. Over the last 3 years, we've generated terabytes of information, data sets across multiple parameters that includes tropism, manufacturability, translatability, as well as the immune phenotype of these capsids. We can use these SARs both to design libraries that are functionally favorably biased for these attributes, as well as now doing training algorithms to do generative design as well. When we design these capsids, we work both on the AAV backbone as well as peptide inserts. When we do both these 2 things, it gives us the most diverse genotypic capsid, as well as the best chance of improving over AAV9 or other conventional serotypes as well. At the same time, we also do protein engineering, meaning we can now structurally design these capsids with how they dock to the putative receptors, including not just NHP, but also humans as well. The 4 dimensions that we work on are in parallel parameters: tropism, manufacturability, immune phenotype, and translatability. For tropism, we do our screens in NHPs, but it also in other species as well, because we want to look for translatability across species and into human. We have a short list of CNS targets that help us cross the blood-brain barrier, where we do in vitro screening, and as well as structural design that I mentioned. These capsids are all manufactured when we design the libraries, meaning not only do we get a yes or no for competence for assembly, but we get quantitative data for upstream harvest, downstream recovery, and packaging efficiency as well. We have a multiplex assay for determining pre-existing immunity in humans with human sera samples. We characterize the capsids that come out also for complement activation, an area of liability for conventional capsids. And last but not least, in fact, the most important, we're quick to pair up our winning capsids with therapeutic cargo, because at the end, this is medicines that need to go in the clinic and to help patients. Since we formed the company, it's been about 3 years, and the improvement we've made with AAV has been phenomenal. AAV9 has been good for some diseases, but we believe limiting, unfortunately, for many other prevalent diseases. The technology that we in-licensed was from the lab of Luc Vandenberg. Luc, as many of you know, is a pioneer in AAV. With the lab work that he had done and the technology that we in-licensed, we were actually pleased at the time with what we had in hand. AAV9 unfortunately only transduces about 1% or less of neurons. And in skeletal muscle, it's in the single-digit % of myocytes that it transduces. With the capsids we in-licensed, with ANK-ADL65, we were at about 5%. And with ATV0024, we significantly detarget the liver. Since then, our N series of capsids targets more than 60% of neurons, and our M series of capsids targets more than 50% of both cardiomyocytes as well as skeletal muscle myocytes. This has the potential to unlock many different diseases, both for our internal pipeline, but also for potential partnerships as well. To walk you through a little bit of some of the capsids that we have in hand. This is an NHP study with a set of pooled capsids. The y-axis is fold improvement over AAV9. This is mRNA enrichment of the barcode, and it's a log scale. The x-axis is different tissues, starting with the cortical regions in the CNS initially, the deeper brain CNS regions in the middle, and then on the very right, two tissues that you would like to detarget, liver and dorsal root ganglia. You can see this series of capsids looking just at N1 through N4 has 2 to 3 orders of magnitude expression improvement over AAV9, while at the same time detargeting both liver and DRG. We've taken one of these capsids as a representation of this series and done a head-to-head study versus AAV9. Again, this is in adult cynos, NHPs. This study is at a very low dose, E13 VGs per kilogram range. Again, to put it into context, Zolgensma was given at a 1 E14 VGs per kilogram range. At the bottom row there is AAV9, and you can barely see any pink. This is an in situ hybridization with pink representing an undisclosed transgene, a therapeutic transgene. This is unfortunately the case for AAV9 at a dose that is in the E13 VGs per kilogram range. Less than 1% of neurons are transduced, and if anything, barely something you can see. With the N1 capsid representing one in this series of capsids, you can see significant amount of ISH staining pink over there. And if you look at the percent of neurons and astrocytes that are transduced, you can see about more than half of them are transduced. The first 3 columns are 3 different, quite relevant regions of the cortex for many different diseases. The 4th column is basal ganglia. The principal reason why you have less pink on the basal ganglia is one of biology and anatomy. The deep brain regions have significantly less, a fraction of the neurons that are present in cortical regions. We then paired up this ISH that I showed you on the previous slide with double staining IHC. The pink here, again, is the undisclosed therapeutic transgene, quite prominent both in the 2 cortical regions, representing the first 2 columns there, and the basal ganglia, the deep brain region on the right there. The green staining there is cellular processes, so principally cells. The 3 insets that you see there represent nu-N for neurons and then GFAP for astrocytes, showing that a significant percent of the cells that are transduced and expressing over here, both mRNA and protein, are neurons and astrocytes, which is very relevant for multiple diseases. We've done the same amount of work for muscle capsids as well, both in cardiomyocytes and in skeletal muscle myocytes. This, again, is a head-to-head study versus AAV9 in adult cynos and NHPs. This work was done for our collaborator, Vertex. In this particular case, we looked at AAV9 versus 2 capsids that we have developed ourselves using structural modeling, M3 and M1. M1 is the only one that detargets the liver because we put an AAV backbone motif in it. In M3, we just put the myopeptide on it. Again, the y-axis here, in this particular case, the y-axis checks the GFP as a reporter gene. This is the percent of cells that are positive for GFP. The x-axis represents different tissues, starting with the liver, which you want to avoid, the ventricle wall representing potential cardiac diseases that you could target, and then 4 different skeletal muscle regions for multiple different diseases there. The gray bar is AAV9, and then the blue and the green bar are the Affinia novel capsids. Again, if you start with AAV9, you can see much of it goes to the liver. Quite decent goes to the ventricle wall in the heart, especially at this 1E14 dose. But then as you go into the skeletal muscles, you see high single-digit or at most low double-digit % of skeletal myocytes being transduced, so suboptimal. And with both M3 and M1, you see significantly more skeletal muscles being transduced and modestly better than AAV9 in the ventricle wall. With M1, again, notably the one capsid that we did design in the liver detarget, and you can see significantly reduced liver transduction. They say a picture is 1,000 words, so here's the picture. If you start with AAV9, very scanty transduction and expression in the quad, and also not homogeneous. In the heart, you have something better. Again, this is a massive dose. In the liver, you get robust expression with AAV9, unfortunately, representing some of the potential toxicity in humans as well. With M3 and M1, if you look at quads, you can see very homogeneous transduction and expression over there with GFP. With heart, you see a little bit better than AAV9. And then with M1, you can see significantly reduced liver transduction, providing potential safety advantages. So coming to the heart over here, we hypothesized that the main reason why we had modest improvement over AAV9 was primarily because the cardiomyocytes with AAV9 were being saturated. So as a starting point, we did a dose-down study in mouse, which is represented over here. The 4 different columns represent increasing doses. The previous study was NHP and 1E14 VGs per kilogram. Here, the highest dose that we studied, as you can see at the very right, is 2E13 VGs per kilogram, so about an order of magnitude lower. The first 2 rows are our capsids, M1 and M3, and then the row at the very bottom is AAV9. If you start with the bottom right on this slide, AAV9 at the 2E13 VGs per kilogram dose, by the way, this is heart, with the tissue that you see represented there being the myocardium separating the different ventricles. You can see AAV9, as you go lower on the dose, going from the right to the left, unfortunately quickly loses its luster. And then M1 and M3 retain the robust expression in all different dose levels, certainly at the E12 and E13 VGs per kilogram range, representing significant potential dose reduction, just as we reviewed with the CNS capsids as well. When we find these capsids, we are quick, as I mentioned, to look at not only tropism, but multiple other parameters as well. And this shows you a representation of their manufacturability, quality attributes, as well as pre-existing immunity, things that are relevant commercially. The chart on the very left shows you the potential dose reduction in all the structure-activity relationships that we have seen. AAV9 is actually the best producing capsid that there is out there. Over time, we believe we can improve on that. But for the time being, some of these capsids are as good as AAV9 in manufacturing or slightly less. When you compound their current yield with the dose reduction that I showed you from the previous slides in both CNS and muscle, you get profound potential for dose reduction overall, meaning compared with AAV9, if you look at the amount of liters of upstream harvest to fund an approximately 70 kilogram patient, we're about half of what AAV9 requires. We not only look at yields, but we also look at quality attributes. The panel in the middle shows you packaging efficiency, and you can see that for our new capsids, predominantly we package more than 90% of full capsids, just as AAV9 does. And then the last panel there shows you, in 2 different studies, human donor samples with pre-existing immunity. And again, here too, we are just as good as AAV9. Worth noting that these capsids were screened for pre-existing immunity. We have not optimized against them, meaning over time, if we wish to, we could actually improve on this from a pre-existing immunity perspective as well. We are in the midst of preclinical studies to determine our own pipeline. We look at 2 things when we look at the indication we want to select. One is the high value potential and market size of the indications. And then the second is the probability of success towards an approved therapy. For high value, obviously, we would like to make the most difference to the largest meaningful number of patients. And then for probability of success, we are evaluating a number of different ways of getting therapy, both into the clinic, but also potential approval, including pathways that might allow us to use biomarker-driven accelerated approval pathways as well. As with any platform company, partnerships are a key part of the company strategy. We have been quite strategic about the partnerships that we select. We have left a principal part of the company unencumbered, for example, in CNS. We're very grateful to Vertex for the partnership that we've done with them since inception of the company. It has a narrow focus. The M1 and M3 capsids that I showed you were done with the work together with them. They have the fields licensed to them for Duchenne muscular dystrophy and myotonic dystrophy. Those capsids are available for us for our own internal pipeline, as well as potential future partnerships as well. And the financials shown on the right are robust. I'm grateful, by the way, to the team that works with me. I mentioned a little bit about my background. The people that have surrounded me include Charlie Albright, who's our CSO. He put CRISPR-Cas9 into the clinic at Editas. Laura Richmond, who's our CDO. She was the executive director at the gene therapy program at Penn, having worked on more than a dozen INDs for gene therapy specifically. And then Rob May, our head of manufacturing, and Petra Kaufman, our Chief Medical Officer, who were together with me at Avexis, getting Zolgensma approved across multiple jurisdictions in the US, Europe, and elsewhere. Appreciate your time and attention. I'm open for questions.